Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today we are going to start with the structure of glucose. Structure of glucose, as you can see in the slide, it can be a straight chain or it can be a closed chain. And it can be an open chain as well as it can be a split closed chain. So the open chain structure was studied by Fisher, okay, and the ring form was studied by Howard. This is known as what? Fisher projection, and this is the Howard projection, right? Now, as you see, glucose is a aldehydic sugar. It is having a aldehydic group, aldose sugar. There are 5 OH groups attached to it, so pentahydroxide. So when we define biochemistry of carbohydrates, then it can also be given as that carbohydrates, they are polyhydroxy derivatives of polyhydroxy derivatives of aldehydes or ketones. They are polyhydroxy derivatives of aldehydes or ketones, right? So according to this structure, Fisher projection, you can see it is a aldose sugar, which is pentahydroxide, which is pentahydroxide, okay, when it is having 5 OH groups attached to it, okay. The first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon, okay. Now, there will be different isomeric forms of this glucose around. We have 16 isomers of glucose, either in the form of Fisher, means straight open chain, or in the form of ring, right? So this ring transformation, I will show you how the anomeric carbon is formed, how the anomeric form is, it is synthesized. Before doing that, I just want you to focus on these 3 positions: the second carbon, the third carbon, and the fourth carbon. These positions of second carbon, third carbon, fourth carbon, they can be the stereo centers as they have chiral carbon atom, mean asymmetric carbon, mean a carbon which is attached to 4 different groups. So when this second carbon, third carbon, and the fourth carbon, the position of OH group and the H group is interchange. If it is change, okay, then it gives what? The different types of relative configurations. Like in case of sugars, we define, like if I take a simplest example of glyceraldehyde, which is a 3 carbon sugar, okay, glyceraldehyde, the simplest sugar, triose. The CHO group, CH2OH group, OH group, and H group. If the OH group is on the right side, then it is the capital D form. And if the OH group is on the left side, then it is L form. Okay, capital D and capital L, they are relative configurations. They are not optical isomerized products like dextro or levo. Capital D and capital L doesn't mean dextro or levo. They are the relative configurations. They are just saying that the OH group is present on which side of the plane. Okay, capital D form and the capital L form. Dextro and levo, they are the optical isomers of these compounds. Mean when these compounds they are passed through the plane polarized light, so they turn the plane polarized light either to the right side or into the left side. Right side, if they turn the plane of the polarized light to the right side, then it is the dextrorotation. And if they turn the plane to the left side, then it is levorotation. Dextrorotation, it is denoted by plus, and levorotation by minus. Now, the capital form the capital form of D is the relative configuration, and the small form of the D is the dextro. Please remember, small d, small l, dextro levo, capital D, capital L, relative configurations. Understood? So these are given for the for they are specially given. These are the specific notations given for the carbohydrates. Okay, the relative configurations. Understood? Now, anomeric form of glucose. How this open chain comes to form a closed chain. Okay, you can see this is the carbonyl carbon. Now this carbonyl carbon undergoes nucleophilic addition. Okay, it undergoes addition. So this is first carbon, second carbon, third carbon, fourth carbon, fifth carbon. So the OH of this fifth carbon will add on the first carbon. So when OH of the fifth carbon will add on the first carbon, then what will happen? The bond, the double bond will break. Okay, this double bond will break. Or I'll show you simply. This is C double bond O H. This is CH2OH. This is the fifth carbon. Okay, OH. I'm not making the other hydroxyl groups. Now you have to attach this OH group to this carbon. So what you have to do, you have to break the bond. So shift the electrons to the oxygen, minus, plus. The lone pairs will be given to this carbon. When the lone pair will be given to this carbon, the OH will attach on this carbon. And these two carbons, they will bond with this oxygen. How? Look here. OH is attached to first carbon. This is H. CH2OH, like this. So you can see the fifth carbon, OH is absent. Fifth carbon and first carbon, they are attached with the oxygen. So when two carbon are attached with the oxygen, then it is known as what? Ether. So this is known as what? Ethereal linkage. Okay. Or anomers. They are known as what? Anomers. Okay. So rest I will make the other OH, H, OH, H. 1, 2, 3, 4, 5 and 6. So this is the anomeric form. This is a ring, this is the closed or a ring form. Okay. Now, when this ring form is represented in the form of pyranose ring, then it is known as what? Glucopyranose. Now, these ring forms are of two types, alpha form and the beta form. These ring forms are of two types, alpha form and beta form. Okay. This is the alpha form and this is the beta form. Now the alpha form is, you can see the anomeric oxygen, this oxygen. If the anomeric oxygen, this oxygen is having the OH group on the first carbon, OH group on the first carbon, you can see the OH group on the first carbon is on the right hand side. Here in the example I have drawn, the anomeric oxygen is this oxygen and the OH group is on the right hand side. OH group on the right hand side on the first carbon. Then it is known as what? The alpha form. Alpha form. Okay. And if I make the beta form, then it comes on the this side. Right? Understood? So this is the beta form. So the rings forms they are categorized into two, the beta form and the alpha form. The alpha form is when the anomeric oxygen OH is on the right hand side. When the anomeric oxygen OH is on the left hand side, then it is beta form. Now in the pyranose structure, it is written what? It is written as what? The groups which are attached on the right hand side will be down to the plane, and the groups which are on the left hand side will be above the plane of the paper. Like for example, OH is on the right hand side, then it is below the plane, and H which is on the left hand side is above the plane. Okay, same here. Second carbon, OH on the right hand side below the plane, H on the left hand side above the plane. So this is what the alpha glucopyranose, which is 38%. Beta glucopyranose is 62%. So beta form is more prominent. Okay, in case of glucose. So this is the anomeric form, the epimers, the alpha and the beta form, the D and the L configurations. Okay. Now, this is pyranose structure and the furanose structure. Furanose structure, you can see the fructose. It is a ketonic sugar. RCOR ketone group is here. Okay. So same thing, the OH will add to the carbon, to this carbon and the fifth carbon. So the second carbon and the fifth carbon, they will be having the this anomeric linkage or ethereal oxygen linkage. So you can see the second carbon and the fifth carbon, they have anomeric linkage. First carbon is above the plane of the paper. Sixth carbon is also the above the plane of the paper. Okay. Right hand side down, left hand side up. Is that understood? This is the fructofuranose. Now, this is deoxyribose. The second carbon is not having oxygen. That is why it is known as what? 2-deoxyribose. While in case of ribose, everything is same. But on the second carbon, which is present. On the second carbon, OH is present. This is ribose. Okay? Understood? Yeah. The saccharides have either ring or straight chain structure. Epimers I have already explained. When we change the positions of OH group, H group on the second, third, and fourth carbon atom, then it is known as what? The epimers. Okay. Mannose is an epimer of glucose when we are having 16 isomers in total of glucose. Mannose is the epimer of glucose, which is having a difference of second carbon. Galactose is also epimer, which is having the arrangement of OH and H differently done on the fourth carbon. Okay. So these are the examples of monosaccharides. Trioses having 3 carbon atoms. Common formula C3H6O3 as a general formula is CH2O whole N. So C3H6O3. Okay. Examples glyceraldehyde and dihydroxyacetone. Dihydroxyacetone among the monosaccharides and glyceraldehyde are the examples of triose family. Tetroses 4 carbon C4H8O4 erythrose and erythrulose. Okay. Dihydroxyacetone doesn't show the chiral carbon. It doesn't show the optical activity. Please remember that also. Pentoses 5 carbon C5H10O5 xylose, xylulose, deoxyribose, ribose, ribulose. All are monosaccharides. Hexose is 6 carbon ramenose, mannose, galactose, glucose, fructose. And haptose is 7 carbon pseudo. So this is the epimers. You can see the glucose, D-glucose, OH on the right hand side you can see. Or you remember like this, that this carbon, please stick to this arrangement. If the CH2OH and the OH are on the same plane, on the right hand side, then it is D-glucose. If on the left hand side, then L-glucose. Okay. So this is the second carbon. You can see the OH on the second carbon on the left hand side, here right hand side. Okay, so this is what the epimers at carbon 2. And at carbon number 4, you can see right hand side, left hand side. That is also epimer. So galactose, the epimer at C4 position, and mannose, the epimer at C2 position. Examples. Ribulose, it is found in nucleoplasm. Okay, nucleoplasm. Arabinose, it is found in gum arabic, which is obtained from organisms like Acacia arabia. Okay, Acacia senegal. So arabinose, it is found in gum arabic, which is obtained from organisms like Acacia arabia or Acacia senegal. Common use in cold drinks. It is used in cold drinks. Arabinose is used in cold drinks. Okay. Xylose, it is found in cell wall of plants. It is found in cell wall of plants. Xylulose, it is a component of hemicellulose in the wood of the plants. So xylose is found in the cell wall of the plants, and xylulose is a component of hemicellulose in the woods of the plants. Glucose is found in high amount in grapes, thus it is known as what grape sugar, high levels in blood and thus called as blood sugar. Forms main energy source and it's a respiratory substrate in the cell. The mirror image is also called as dextrose. So please remember this, that the mirror image of glucose structural wise is known as what? Dextrose. Okay. Fructose has a sweetest taste, present in high amounts in honey, sweet fruits, and thus termed as fruit sugar. So glucose, grape sugar, fructose, the fruit sugar. Thematin is the sweetest carbohydrate. Thematin is the sweetest carbohydrate, which is extracted from Tomatococcus dandeli bacteria. Aspartame is commonly used as artificial sweetener in most of the foods. The thematin is the most sweetest carbohydrate which is extracted from Tomatococcus dandeli bacteria, and aspartame or aspartin, it is a artificial sweetener which is used as a sweetening agent, and it is non-carcinogenic. Okay, aspartame is a non-carcinogenic. Okay. Galactose is the brain sugar. It is another name given to the galactose, brain sugar, because it is utilized in high amounts in brain and nervous tissue. This sugar is always a part of some compound, never present in free form. Okay, like hemicellulose, lactose, pectin, glycolipid. Okay, so galactose is also this brain sugar. It is always found attached to the some other compounds. It never exists in free form. Example: hemicellulose, glycolipids, pectin, lactose. Mannose, even this sugar is accompanied with some other compound and not available in free form. Mannose. Mannose is found in albumin of egg, hemicellulose of wood. Okay. Rhamnose, the second carbon atom in the structure lacks one oxygen atom. and the molecular formula is C6H12O5. The second carbon is not having oxygen, okay? So it is also known as 2-deoxy or 2-anoxy. The sugar is present in phloem. Ribose is present in phloem. Mannose is present in albumin, egg, hemicellulose of wood. Okay. Galactose is present in brain and nervous tissue. Hemicellulose, lactose, pectin, glycolipid. Derivatives. These are the derivatives of monosaccharides like amino sugars, sugar alcohol, sugar acids. Amino sugars, they are the hydroxyl group in the second carbon atom, which is replaced by the amino group. So the hydroxyl group, OH group, is replaced by NH2 group. OH group is replaced by NH2 group. So glucosamine, galactosamine, they are the amino sugars. Glucosamine and galactosamine, they are amino sugars. Sugar alcohol, the aldehydic group CHO in the sugar is displaced by primary alcohol CH2OH like sorbitol and mannitol. It basically comes when we oxidize the glucose or sugars. When these sugars are oxidized, 2, 3, 4, 5, and 6. So when they are oxidized with mild oxidizing agent, okay. So when they are oxidized with mild oxidizing agent, they form sugar acids, okay, like glucuronic acid and galactornic acid or saccharinic acid. Saccharinic acid. Like if the CHO group is oxidized to COOH on one side, then it is glucuronic acid, okay. Like if the structure is this, everything is remains same. It is same. But if the CH2OH, the primary alcoholic group part, is also oxidized to COOH, then it is saccharinic acid. Okay? Then it is saccharinic acid. So when they are oxidized with mild oxidizing agents or strong oxidizing agent like HNO3, then it is saccharinic acid. Mild oxidizing agent like bromine water, then it is the glucuronic acid. When they are reduced, okay? So when they are reduced, they are known as what? The alcoholic part or alcoholic sugars, sorbitol and mannitol. So amino sugars, glucosamine, galactosamine, alcoholic sugars, sorbitol, mannitol, okay, and sugar acids, glucuronic acid, saccharinic acid, and galactornic acid. Understood? What are reducing sugars and what are non-reducing sugars? Reducing sugars are carbohydrates that can act as a reducing agent due to the presence of free aldehydic or ketonic group. If any sugar is having free aldehydic or ketonic group, so almost all monosaccharides, they are reducing. Almost all monosaccharides are reducing because they have free aldehydic and ketonic group. In case of disaccharides, when we talk about the disaccharide of glucose and fructose, that is sucrose, it is non-reducing. Non-reducing sugars are the carbohydrates that cannot act as a reducing agent because they don't have absence or they don't have presence of free aldehydic or ketonic group. They are good reducing agents. They are not reducing agents. They have free aldehydic group, ketonic group. They don't have free aldehydic or ketonic group. Now, how will you confirm or how will we confirm the presence of aldehydic or ketonic group by certain tests like Felling's test, Benedict solution? Okay. So Felling test It includes phelling A and phelling B. In phelling A and phelling B, we have a mixture of aqueous copper sulfate and Rochelle salt. When we mix both phelling A and phelling B, we get a mixture of aqueous copper sulfate and Rochelle salt. So, if these sugars are reducing, then they reduce the copper sulfate Cu2 plus ions into metallic copper, or they form the copper oxide, either metallic copper or cuprous oxide or cupric oxide. Your red PPT, you will get a red PPT. Metallic copper also red precipitate. Understood? Yeah. So they gives a positive reaction towards the Fehling test. They gives a negative reaction towards Fehling test. So if the sugar is reducing, it will reduce the, it will act as a reducing agent. It itself will oxidize, the aldehyde group will oxidize to the acidic group, and they will reduce the metallic, the copper ions from the CuSO4 to Cu metal. Include all monosaccharides and some disaccharides. Okay, in case of disaccharides, sucrose is non-reducing. Okay, because in case of sucrose, the free aldehydic and ketonic group, they are bond together to form glycosidic linkage. Because free aldehydic or ketonic group is utilized to form glycosidic linkage. Okay, understood? Now we are going to start with oligosaccharides. What are oligosaccharides? Oligosaccharides general characteristic: the carbohydrates on hydrolysis give 2 to 10 monosaccharides. So oligosaccharides, they are formed by the glycosidic linkage, by the glycosidic bond. Okay, so when sugars or two saccharide or 2 to 10 monosaccharide units they combine, they form the oligosaccharide. So these monosaccharides, they are known as what? Monomers. Okay. The monosaccharides have glycosidic bonds that bind them together. The glycosidic bond is formed when aldehyde or ketone group of one monosaccharide reacts with the alcoholic group of the another monosaccharide. So please remember, the glycosidic bond is the bond between the aldehydic group of one monosaccharide to the alcoholic group of the another monosaccharide. The structure loses one molecule of H2O, so it is a condensation reaction. It is a type of condensation reaction which involves the removal of or the elimination of simple molecules. So the structure loses one molecule of water during the glycosidic bond formation, dehydration, synthesis. Okay. So what are the general characteristics of oligosaccharides? Oligosaccharides, the carbohydrates on hydrolysis gives 2 to 10 monosaccharide units, monomers, and are called as oligosaccharides. The monosaccharides have glycosidic linkage. The glycosidic bond is formed between the aldehydic group of aldehydic or ketonic group of one monosaccharide to the alcoholic group of another monosaccharide. The structure loses one molecule of H2O, so it is a dehydration reaction or a condensation reaction. Is it clear? Now, types of oligosaccharides. Disaccharide, in which two saccharide units are there, has two. So please remember, there is a difference between trisaccharide and triose. Triose is an example of monosaccharide sugar, and trisaccharide is having three saccharide units, three sugar units. Okay, so disaccharide having two monosaccharide units in the structure like maltose, sucrose, lactose, terhulose. Okay, so maltose, sucrose, lactose, and trehalose, They are the examples of disaccharides. Trehalose is found in some of the fungi and hemolymph of some insects. Okay, so please remember, trehalose is found in some of the fungi and found in some insects. All the disaccharides are water soluble. They are water soluble and taste sweet. Thus, they are called as sugar. All the disaccharides, they are water soluble and they are having sweet taste. That is why they use what? Sugar. Maltose. Maltose is known as what? Malt sugar, which is an intermediate compound in the starch digestion by enzymes. When there is enzymatic breakdown of starch, then maltose is an intermediate product. Okay. Maltose has 1,4 glycosidic linkage. That means what? In case of maltose, the first carbon of one of the monosaccharide makes the glycosidic bond with the fourth carbon of the another monosaccharide. So they have the 1,4 glycosidic linkage. They have 1,4 glycosidic linkage between alpha D glucose and alpha D glucose. So maltose is a reducing sugar. So maltose shows alpha 1,4 glycosidic linkage and they have free aldehyde group, that is why they are reducing in nature. They have free aldehyde group, that is why they are reducing in nature. Lactose is a milk sugar with beta 1,4 glycosidic linkage between beta D glucose and beta D galactose. So lactose is also a reducing sugar. Maltose and lactose both are reducing sugar because both of them they are having free aldehyde group. Maltose is having alpha 1,4 glycosidic linkage and beta and that lactose is having the beta 1,4 glycosidic linkage. Lactose is the milk sugar and maltose is the malt sugar. Okay, lactose is almost tasteless or very less sweet. Human milk contains maximum lactose, which is around 7%. The sugar in plants is transported in the form of sucrose. Okay, you know about the Munch flow hypothesis? Okay, so these are some common oligosaccharides like disaccharides. In disaccharides you have to remember about maltose and lactose, alpha 1,4 glycosidic linkage, beta 1,4 glycosidic linkage. Okay, they are reducing sugars. Lactose is also a reducing sugar. Now, there is a term which is known as what? Soaring of milk. Soaring of milk is basically because of the conversion of lactose into lactic acid. Okay, these are disaccharides. Then sucrose is also called by many names. Sucrose is a disaccharide which is called by many names. Okay, like invert sugar, cane sugar, table sugar, or common sugar or commercial sugar. The sucrose is also a disaccharide. Understood? Then sucrose is made up of alpha D glucose and beta D fructose. The sucrose is made up of alpha D glucose and beta D fructose, and that is why it is a non-reducing sugar. Okay, non-reducing sugar because no free aldehyde or ketonic. The hemolymph of insects is trehalose. There is a glycosidic linkage between two anomeric carbon atoms like D glucose and E glucose. Okay, so the hemolymph of insects is trehalose. Hemolymph of insects is having trehalose. There is a glycosidic linkage between two anomeric carbons. There is a glycosidic linkage always between two anomeric carbons. Okay, of D glucose and D. It is not E glucose. It is E. D glucose and D glucose or 1,1 linkage. Thus, trehalose is a non-reducing sugar. Okay. So trehalose is also a non-reducing sugar, okay, because it is having a 1,1 linkage. Because of the first anomeric carbon, only the aldehyde group is there, and if it is involved, then it is a non-reducing sugar. Now, raffinose is also a reducing sugar. It is formed of glucose, fructose, and galactose. It is a, you know, trisaccharide. Trisaccharide has three monosaccharide units in the structure like raffinose, which is galactose, glucose, and fructose. So galactose, glucose, and fructose is raffinose, and it is a reducing sugar. Tetrasaccharide, four monosaccharide units in the structure. Stachyose, stachyose is having galactose plus glucose plus fructose. Okay, so stachyose is also a tetrasaccharide having galactose, glucose plus fructose. Understood? Pentasaccharide, five monosaccharide units in the structure like barbascose. Barbascose is having galactose, glucose, fructose, okay, and some other unit which is missing. So it is having a five monosaccharide units, barbascose, galactose plus glucose plus fructose, and one another unit is missing. Raffinose, stachyose are present in phloem cells in plants and also be used for the translocation of carbohydrates in phloem. So raffinose and stachyose they are present in phloem cells and they help in the transportation of or they help in the translocation of food through phloem. Understood? Raffinose and stachyose. Okay, is that clear? So trisaccharides, tetrasaccharides, pentasaccharides, and raffinose and stachyose, they are present in phloem cells in plants and they can also be used for the translocation of carbohydrates and phloem. Now, there are many more uses of these, we can say, the oligosaccharides, like to form glycocalyx or the cell coat. So, there are the structural formula of some disaccharides, alpha 1 for glycosidic linkage. You can see this is the anomeric carbon, but this carbon is not anomeric. Okay, so this carbon, which is anomeric, is involving its group. Okay, to attach with the hydroxyl group, so the aldehydic group attached to the hydroxyl group. Okay, with the other carbon, maltose or alpha 1,4 glycosidic linkage. Okay, this is maltose, this is lactose. Okay, so I was telling the uses like economic importance. Okay, the oligosaccharides attached to the cell membranes. Oligosaccharides they help to form cell coat or glycocalyx. They help in immune processes like cell recognition, cell attachment, receptor molecules, antigen specificity, and many more. Specificity. Okay. To make glycoproteins, so these are the economic importance of oligosaccharides to make cell coat, glycocalyx. They help in recognition of foreign bodies on the cell or foreign cell recognition, cell attachment, cell to cell attachment, receptor molecules, antigen specificity, and to make glycoproteins. Okay, understood? Oligosaccharides they also act as a reserve food material. Okay, like in case of sugarcane and sugar beet in the form of sucrose. Okay. Some of the oligosaccharides like ribose, okay, they are very important to make the nucleic acids, to make the metabolic precursors or the flavoproteins, okay, to make metabolic precursors metabolic precursors. Okay, and to make coenzymes. Okay, like coenzyme A, flavin adenine dinucleotide, nicotinamide adenine dinucleotide. Okay, RNA, DNA, monosaccharides. Understood. So they all are the economic importance. Like ribulose 1,5 biphosphate is a phosphorylated pentose sugar. Okay, monosaccharide, which is the most abundant sugar of this organic world. Okay, so all these are the examples, the structures of disaccharides, monosaccharides. Okay, this is sucrose alpha 1 comma 2 glycosidic linkage. So you can see 1 comma 2 glycosidic linkage. Basically, the free aldehydic group is involved in the glycosidic formation. That is why it is not a reducing sugar. Polysaccharides. Polysaccharides, the general characteristics are polysaccharides are made up of large number of monosaccharide units. The names end in a suffix n so that they are known as glycans. Okay. The polysaccharides, they are made up of large number of monosaccharide units, means they are the polymers. They are the polymers. Okay. Pentose polysaccharide are commonly termed as pentogens. For example, arabin from L-arabinose, xylene from D-xylose present in cell wall. Okay. So pentose polysaccharides are commonly termed as pentogens. For example, arabin from L-arabinose, xylene from D-xylose present in cell wall. Hexose polysaccharides are called as hexans. For example, mannans from mannose, cellulose, starch, etc. present in plants and animals. Hexose polysaccharides are called hexans. For example, mannans from mannose, cellulose, okay, comma starch present in plants and animals. Polysaccharides are non-soluble in water, non-reducing, and taste sweet but less. Polysaccharides, they are non-soluble in water because of their giant size, non-reducing, and they have taste when sweetless. Their taste is sweetless. They are classified as nutritive and structural polysaccharides on the basis of their functional group. Because of their structure, they are classified as nutritive and structural polysaccharides. So polysaccharides, they are the polymers of monosaccharides, okay, and their names are added with the suffix an. For example, a polysaccharide of 5 carbon sugar pentose is pentosan. Example, arabin, which is obtained from L-arabinose, xylene from D-xylose, okay, which are present in cell wall. Hexose polysaccharides like hexanes from mannan, from mannose, cellulose starch from plants and animals. Polysaccharides, they are non-soluble in water, non-reducing and tasteless. Okay, they are classified as nutritive and structural polysaccharides on the basis of their functional groups. Types of polysaccharides: homopolysaccharides and heteropolysaccharides. Homopolysaccharides, they have some monomers in their structures. Okay, they have same monomers in their structures. Sorry, they have same monomers in their structures. The important ones in terms of biology are as follows: cellulose. Cellulose is a linear polymer of beta D glucose units. Okay, 6,000 to 10,000 daltons of molecular weight, which have beta 1, 4 linkage among the glucose molecules. When the glucose is partially digested, it gives us cellobiose unit, which is a disaccharide. So cellulose is a linear polymer of beta D glucose, 6,000 to 12,000, 10,000 molecular weight of daltons of molecular weight, which have 1, 4 beta 1, 4 glycosidic linkage. Okay. When the cellulose is partially digested, it forms or it gives cellobiose, which is a disaccharide. Cellulose forms major component in plant cell. Cellulose are 50% in wood and 90% in cotton. It is most abundant molecule for its organic matter on earth. Eukaryotes have cellulose-like material called tunicin, which is also called as animal cellulose. Eukaryotes have cellulose-like material known as what? Tunicin, which is also called as animal cellulose. It is used in the manufacturing of rayon fiber. Cellulose is used in the manufacturing of rayon fiber, which is artificial silk, as it is a semi-synthetic fiber. Okay. So homopolysaccharides, they having same polymers, heteropolysaccharides, different polymers. Like for example, cellulose, which is a linear polymer of beta D glucose units, molecular weight 6,000 to 10,000 daltons, which have beta 1,4 glycosidic linkage. The molecules of glucose are arranged in a straight chain, and they combine with the glycosidic linkage. There is no branching. When it is partially digested, it forms cellobiomes. Tunicin is the cellulose-like material secreted by uropodates, which is also called as animal cellulose. Now, starch, these are the different starch grains you can see. Okay, starch is also a polymer of is a polysaccharide. Means it is a polymer. Understood? So, starch is a storage polysaccharide of most plants. It is a storage polysaccharide. So, starch is the storage polysaccharide of plants. Okay. In human beings they obtain starch from different food products like rice, wheat, legumes, okay, banana. It is a polyglucan homopolysaccharide. It is also a homopolysaccharide. Polyglucan. Okay, understood? These are the different starch grains. Okay, you can see that the simple starch grains of potato. These are the simple starch grains of potato. In case of potato, starch grains of potato. Compound starch grains of potato is also given. Okay, when these simple grains they join to form, like they join to form like this structure. So these are compound starch grains. Compound starch grains. Okay. Then the simple starch grains of maize, you can see simple starch grains of maize. So these are the different starch grains of maize, banana, ठीक है ना? Rice, wheat, pea, all these are the starch grains. Understood? Hilum is the precipitance of the these shells or the layers which are arranged in the concentric or eccentric fashion around a proteinaceous point. It is known as what? Hilum. Okay. So hilum is basically what the arrangement of the layers or the shells or layers around the proteinaceous point is known as what? Hilum. Okay. Starch. Starch is a storage food of carbohydrate in the plant. Starch is a polymer of alpha D glucose units. Starch consists of two types of chains, amylose and amylopectin. Amylose is the unbranched polymer with 250 to 300 glucose units joined with alpha 1,4 glycosidic linkage, and amylopectin is a branch chain of 30 glucose units that are linked with alpha 1,4 to alpha 1, and alpha 1,6 linkage bonds. So alpha 1,4 is for straight and alpha 1,6 for branch. So amylose and amylopectin. Amylose is the unbranched polymer with 250 to 30, 350 glucose units, and amylopectin is a branch of 30 glucose units that are linked with alpha 1,4 and alpha 1,6 glycosidic linkage. Okay. Amylose with iodine. Amylose with iodine gives blue color, while amylopectin gives red color. Okay. Amylose with iodine gives blue color, and amylopectin gives red color. Starch contains 20% amylose and 80% amylopectin, which is present in potato. Potato starch turns purple or violet in color when mixed with iodine because of the mixing of red and blue. Understood. Amylose with iodine gives blue color with amylopectin and red color with red color with amylopectin, blue color with amylose. Okay, starch contains 20% amylose and 80% amylopectin, which is present in potato. Potato starch turns purple or violet in color when mixed with iodine because of the intermixing of red and blue color. Understood? So this is about the polysaccharides of starch, like starch and cellulose. We will be discussing more in the next turn. Till then, bye bye. Take care. Stay safe. Stay happy.